Hello, I'm Julie Theriot. I'm a professor at Stanford University. And for the second part of my iBio Seminars presentation today, I'd like to delve into the details of the mechanics and dynamics of rapid cell motility. Now, to get us started here, we see two images of a particular kind of very rapidly moving cell that comes from the skin of fish called a keratocyte. Over on the left, we have a fixed cell that's been labeled for filamentous actin, so you can see the distribution of the actin cytoskeleton throughout the cell. On the right, we have a video image of the same kind of cell moving in the same direction that the fixed cell was at the time that it was frozen with formaldehyde. And what I'd like to talk about particularly is the way that all of the different molecular machines that have to operate in the context of a moving cell are able to coordinate with one another in order to give this incredibly smooth and elegant gliding motion that you see in rapidly moving cells. So focusing a little bit on the details of these molecular machines, these are things that have actually been intensively studied by biochemists and cell biologists for many decades. And at this point, we're pretty familiar with many of the proteins that are necessary and to some extent even maybe sufficient for generating the forces and dynamics that we see associated with cell motility. And in general, for large-scale cell biological processes like motility, the proteins within the cell that are responsible for that kind of behavior arrange themselves into nanomachines, where a number of different proteins will work together. Now, one nanomachine that's very important in cell motility is the assembly of branched actin structures at the leading edge of a motile cell. And in my in the first part of this presentation, I talked in some detail about how these different components have been identified and how they're thought to work together. But this particular little nanomachine of growing actin filaments pushing against the membrane really only operates in this very front part of the cell, just a few microns back from the plasma membrane at the very leading edge. Another nanomachine that's also been quite intensively studied and is very important for motility is shown here. This is the adhesion that actually binds the cell to the substrate, enabling it to generate traction so that it can move itself forward. And here, too, a lot of the protein components have been identified. And in this particularly beautiful example from Claire Waterman's lab, even the spatial orientation of all the different components have been very carefully measured with respect to one another. But this particular nanomachine that makes this well-organized adhesion really operates only in, again, a very small zone right at the back of the cell. And in order to understand the overall process of cell motility, when the cell is moving forward, it's incredible how well all of these things seem to be coordinated with one another. The cell looks like it's gliding over the substrate without changing shape, even though it's got to be assembling actin filaments like mad at the leading edge in order to push that membrane forward. And then it's got to be building and then disassembling the adhesions at the rear. All of these things have to be coordinated to happen at the same pace so that the left side of the cell moves at the same rate as the right side of the cell so it can go straight, so that the front extends at exactly the same rate that the back retracts so that it can appear to move forward without changing its size. So it's a really fundamental and interesting question, I think, about how all of these different nanomachines can coordinate with one another over the entire span of the cell, which is tens of microns across. So many orders of magnitude larger than certainly the individual proteins that make up the machines, but even any of these individual assemblies on their own. Now, in order to address these kinds of questions about how you get large-scale coordination in moving cells, we've taken a lot of advantage of these fish skin cells. So I'd just like to give you a little bit of background about where they come from and how we isolate them. It turns out that pretty much all fish and many other amphibians have a bilayered epidermis. And the basal layer of that epidermis is made up of these cells that seem to be specialized for very rapid wound healing. So in the context of a fish, when there are scales coming out of the flank of the fish, the epidermis actually wraps around the scale. So if you go in with a pair of blunt forceps and pluck a scale off of a fish and then put it in culture, the scale comes along with just a little bit of skin right along the edge. Now, the fish is not happy about this, but it can grow a new scale, and actually turning over the scales is part of its normal skin regenerative process. So it doesn't cause any significant damage. But in the meantime, in culture now, we have this scale with a little bit of skin that's just wrapped around the tip of it, and the cells at the edge of that little bit of tissue essentially think that the surface of the fish has been wounded. And so their response is to try to start crawling outward to close that gap. And you can see here, both at the bottom and at the top of this particular scale, these big clumps of cells that start coming off first as epithelial sheets, and then eventually break up to make all these little individual cells that seem to go buzzing around more or less on their own. Now here we can look at that same process at higher magnification. Here at the edge of a sheet as it first starts coming off. And then here again, this is what the isolated cells look like once they break away from that epithelium. And I hope you can appreciate from looking at these movies why these cells are such a spectacular model system for studying the mechanics of cell motility. They move extremely fast. They're among the fastest animal cells that are known. And they also have this very characteristic stereotype geometry, which is very well illustrated by the cell over here. It has a very large, flat, broad lamellipodium, which is its motile organ. And then it carries all the rest of its organelles, its nucleus, its Golgi apparatus, even all its microtubules. It carries in this little package of a cell body that it just keeps right behind it. The movement is very fast. The movement is very persistent. The movement is essentially unidirectional. That is, they don't really have a very strong tendency to change. So they're essentially moving at steady state. The fact that the movement is so regular and is so stereotyped makes it very good for biophysical analyses of the kind that I'm going to be delving into today. This shows you a little bit more detail about how the cytoskeleton is organized in these particular cells. And in this beautiful structured illumination micrograph taken by my student Sunny Lowe, you can see the distribution of actin filaments shown in green, myosin 2 filaments, that is contractile myosin, shown in red. And we're going to come back to the role of myosin in this coordination quite a lot. And then also you can see labeled in blue the focal adhesions that are adhering the cell to its substrate. And diagrammatically, shown down here, looking at the cell from the top, you can see the actin filament branch network is primarily oriented towards the front of the cell. And then in the back of the cell down here at the bottom, you can see the filaments have become rearranged to form these parallel bundles that are being organized by the myosin. Now, if we take a cross-section through one of these cells and look at it sideways, it looks like a baseball cap, where the lamellipodium is very, very thin and flat, only about 200 nanometers from top to bottom, and then the cell body can rise up several microns high. As these cells move forward, they follow the same general steps of actin-based cell motility that are shared by many other motile animal cells and also a large number of eukaryotic unicellular organisms such as amoeba. Overall, the first thing that has to happen is the cell has to establish polarity. That is, it has to be able to distinguish its front from its back. And then it has to be able to extend the leading edge. And in this cell, as in many other motile cells, the force for that extension is thought to be driven by actin polymerization itself. As it's extending the new leading edge, it needs to form new adhesions to its substrate and at the same time be able to contract its rear to bring the cell body forward and then retract and disassemble the adhesions that are at the back. One of the fun things about keratocytes is it's actually possible to demonstrate in these cells that all of the components necessary for that whole cycle of motility are contained only in the lamellipodium. You don't actually need any contributions from the cell body. And that was first proved in this really classic 1984 experiment by Ursula Utenauer and Manfred Schliewe, where they sliced little bits off of the lamellipodium of keratocyte, leaving the cell body behind. And we're able to see that those small fragments of the keratocyte lamellipodium were able to continue to translocate on their own and move at just about the same speed and just about as persistently as the whole cell was when it was intact. And this movie here shows a modern reenactment of that experiment that was done by my student Aaron Barnhart. Here you see a fragment that's been isolated away from its cell body that's nothing but lamellipodium with all of these dynamic cytoskeletal structures inside of it. And the movie plays, you can see it's crawling along very nicely. It's got a clear front and a clear rear. It's about to crawl over a little piece of schmutz on the cover slip that actually is going to separate this crawling lamellipodial fragment into two bits. The membrane connection between them resolves, and they're both able to crawl off on their own until they eventually get sliced into some unit that's too small to move. So with the system, we have favorable geometry. It's very, very simple, very reproducible from one cell to another. And we also have a fairly simple self-contained system where we know that it's only the components of the lamellipodium that are necessary for persistent motility. So from analyzing the behavior of these cells over many years, my group has been able to identify and specifically measure the contributions of all of the different force-generating elements that help the cell to move. And those are all illustrated here. At the leading edge, we have actin polymerization that's shown in red, pushes the membrane outward. And that polymerization is actually opposed by tension in the plane of the membrane. And that tension serves both to act as the barrier that the growth of the actin filaments pushes against, and also, in fact, helps to coordinate the motion over the entire surface of the cell, as we'll see in a little bit more detail. Now, there's also adhesions that have to contribute, and those adhesions are assembled at the front and then disassembled at the back. And then there's contractile forces that are driven by myosin, primarily acting at the back. Because that myosin contraction is happening at the back and squeezing the cytoskeleton inward, that actually creates a forward hydrodynamic fluid flow that squirts fluid through the meshwork of the lamellipodium to deliver components up to the front of the leading edge. Now, as I said, we've been able to measure the quantitative contributions of each of these different forces within the context of this very simple kind of motile cell. And what I'd like to do over the next few minutes is share with you a couple of highlights of things that we've learned that are somewhat surprising in retrospect as to how this coordination is able to work over such a large scale. So in order to make these kinds of measurements, we've had to develop methods both for measuring behaviors of the cells very precisely and also methods for perturbing the behaviors of the cells so that we could understand what aspects were dependent on what other aspects. So one example of a kind of measurement that we can make, shown here in a movie from Cyrus Wilson, is tracking of the overall motion of the actin network using a technique called speckle microscopy that was originally developed by Claire Waterman. And in this technique, the cells are electroporated with a small amount of a fluorescent dye that binds to the actin filaments, but a sufficiently small amount that rather than labeling the whole cell uniformly, you instead see this little speckly textured pattern. And then if we take the movies as they come off the microscope, which is what you see up top, and then convert them into a different frame of reference, where instead of looking at the cell in the lab frame of reference, we now translate everything as if the cell had a GoPro camera attached to its head, and we're looking just right down at the cell itself from its own point of view. Then you can see now quite a bit more detail in terms of both in phase contrast and then with this fluorescent speckle microscopy, how everything inside the cell is moving. So looking at the fluorescent speckles, I hope you can now appreciate that the whole actin network is sort of raining downwards from the front towards the back of the cell in the cell's frame of reference, and is also being gathered inwards on the side, down at the back here, where the myosin is able to contract it. Now, once we can do that frame of reference shift and look at things from the cell's point of view, Cyrus Wilson was able to work together with Gaudenz Danuser and people in his lab, including Lin Ji, to develop quantitative methods for measuring the flow of all this material in the lamellipodium very precisely, and was able to map overall how the motion of these particles depended on location inside of the cell. So here from the lab frame of reference, what you can see is that the motion of the particles with respect to the substrate is actually very little. That is, the actin is pretty stationary with respect to the glass that the cell is crawling over, except at the very back where you see this massive inward sweeping driven by myosin. However, if you look from the cell's point of view, you see there's a lot of flux, a lot of turnover of constantly treadmilling actin network, where it's assembling at the front and then disassembling under the cell body. So in order to try to understand this process of assembly and disassembly a little bit better, we also wanted to be able to manipulate the behaviors of the cells, to perturb them so that we could look at how they responded to changes in their environments. And one kind of perturbation that was actually very informative for understanding how all these things were coupled together was worked out by Erin Barnhart, specifically where she was able to change the degree of adhesivity or the degree of stickiness of the substrate that the cells were crawling over. And what she found was when cells are on a sort of moderately sticky substrate, they're able to move exactly the same way that they would on glass or in fact on the surface of an animal. When they were put on substrates that were less sticky, so ones that were more slippery, you can see the cells actually change shape. They become a little bit rounder, and you can see the accumulation of these characteristic pleats in their lamellipodium, where the inward flow of the actin is now actually faster than the motion of the cell. So it's really spinning its wheels because it can't quite get a grip on its surface. Now, most interestingly, I think, when they're put on high adhesion substrates, their behavior changes very dramatically. And instead of now having this steady state motion where they glide forward uniformly, they now do this completely crazy thing of putting out small bits of the lamellipodium that seem to sweep sideways. And we're still in the process of trying to figure out how all of these different things work. But I hope you can appreciate that even this very, very simple motile cell that seems like, sort of the stripped down, minimalized, like soapbox derby version of a motile cell, even this is able to extremely expand its behaviors depending on cues that it's getting from environment. In this particular case, mechanical cues in the form of the stickiness of the substrate. So the several examples that I want to tell you about mostly have to do with surprising roles for myosin. Now, myosin, of course, we're mostly familiar with in the context of skeletal muscle, where it's able to contract sarcomeres by sliding stable arrays of actin filaments relative to one another. Myosin in non-muscle cells, myosin 2 in non-muscle cells, also acts as a contractile protein, and its assembly is regulated. So the monomeric state of the myosin in non-muscle cells is folded up on itself, and then when it receives an appropriate signal, the phosphorylation of the regulatory light chains on myosin enables it to extend outward so that it can then assemble into bipolar thick filaments that are much more similar to the organization inside of muscle. And so when we look at the myosin in keratocytes, what you can see is there's very little myosin at the front where the actin is actively polymerizing, and instead there's actually quite a lot of myosin right at the back. And in particular, it's in these very bright spots right on either side of the cell body. Okay, so bearing all that in mind, now let's go back to this question of assembly and disassembly. One of the things we're able now to measure that we can track the motion of the actin and know where all these other elements are located is Cyrus was able to actually figure out how to make a map of quantitatively how much assembly and disassembly of the actin cytoskeleton takes place over the context of the whole cell. And what he found was that the assembly is very much biased towards the leading edge, specifically right in the middle of the front of the leading edge, which is very much what we'd expect. But the disassembly, unexpectedly, was found in these two very intense spots right on either side of the cell body. And Cyrus recognized that those locations actually were very similar to the locations where we found myosin. Now we can also look at the distribution of myosin 2 in these cells, here using a cell that's been transfected with myosin light chain carrying YFP. And now in this cell frame of reference, you can actually see the motion of these little speckles, which now are mini filaments of myosin, that is bipolar filaments that have been assembled. And what you can see is they seem to stick onto the actin network, and then they rain backwards towards the back of the cell body, essentially riding on the actin network until you get right all the way to the back where they then start forming these contractile cables, pulling in the actin network and making these bundles that go from one side to the other. Now, it's suggestive that the spatial distribution of myosin 2 in these cells is exactly the same as the foci of disassembly. And we can also inhibit disassembly of actin in these cells, for example, by inhibiting the motor activity of myosin. So we hypothesized that the myosin itself is actually contributing to the disassembly of the actin cytoskeleton by buckling and breaking and ripping apart the actin filaments, using directly its force-generating capabilities. One of the strongest pieces of evidence in favor of that hypothesis is this very nice experiment done by Mark Tsushita, where instead of using moving living cells, he used extracted cytoskeletons. So if you take a keratocyte as it's crawling across the substrate and then sneak up on it with a little bit of detergent, you can get the membrane to dissociate, leaving behind only the insoluble parts of the cytoskeleton. So the assembled actin filaments, whatever actin-binding proteins are bound to them, but having now gotten rid of all soluble components, including actin monomers, ATP, everything else. Mark was then able to label those extracted cytoskeletons with phalloidin to see where the actin filaments were, and then add back ATP to those extracted cytoskeletons. That added ATP was able to then activate the myosins that were left behind, so we could see if in this sort of semi-in vitro environment, myosin activity could actually drive destruction of the actin filament network. And so that's what you're going to see in this movie. This is an extracted cytoskeleton now. When the movie starts to play, the ATP is going to be added, and you can see the network just melted right in the back, right where the myosin is located. And you can also see that by comparing this before and after shot, where the blue shows the places that the actin network disappeared when the myosin was activated. So although we normally think of myosin as actually contributing to contraction, in this context, at least, it seems like one of its more important functions is destroying the actin network when it gets to the back of the cell. So putting that together, we came up with this idea for myosin driving overall network treadmilling in the lamellipodium, as illustrated here, where initially, towards the front, there's very little myosin in the network. It's hard for the myosin mini filaments to diffuse through the network as it's actively assembling and essentially pushing everything backwards. But a few of them get a hold of the filaments, and then as they start contracting, they start rearranging the actin filaments to form more parallel structures that are more favorable geometry for force generation by myosin. After that goes on for a while, by the time you get to the back of the cell, the actin is now all in parallel bundles rather than a dendritic network, And the high concentration of myosin that's able to accumulate there over time is enough to rip that network apart. So overall, we think that's a major mechanism for determining what the distance is from the front of the cell to the back of the cell. It's just determined by how much time it takes for myosin to incorporate and for myosin to destroy the network. Now, so far, I've been talking about keratocytes as if they're all exactly identical. And certainly that's one of the useful things about them is that they're similar. But like any other organism, if you look at them closely enough, you'll see they actually have very interesting differences from one another. So this shows a gallery of a whole bunch of different keratocytes that were collected by Theriot, Karen, and Zach Pincus, showing that from even one scale of a particular individual fish, you can have quite a lot of variation, both in terms of the size of the individual cells and then also their shape. So some of them are quite round, and some of them are quite elongated and almost canoe-shaped. And to summarize a lot of work, what we've been able to find is that these cell-to-cell shape differences are both persistent, so if you follow a cell over time, it keeps its shape, and they're also cell-intrinsic. So if you disassemble all the actin cytoskeleton and let it grow back, it'll grow back to exactly the same shape it was before. And from quantitative analysis of those kinds of measurements, we found that these shape differences are essentially extremes of a continuous spectrum, where some cells are very large and wide and smooth, and these are the ones that are canoe-shaped, and those are also the fastest moving cells. And some of the other cells, such as the ones over on the left side of this gallery, are rounder, they tend to be smaller, their leading edges look kind of rough, and they also move very slow and in a less persistent manner. And so we call the wide, smooth cells, we call those coherent cells, and the smaller, narrower, rough cells we call decoherent cells. But overall, we can find every behavior in between. So we think the differences that we see among these shapes of the cells basically just has to do with the exact quantitative amount of all of these different force-generating elements that they have present within their cytoplasm that balance each other in slightly different ways to give different overall cell shapes. And overall, we can quantitatively measure these variations in the cell shape, particularly identifying principal modes of shape variation. And the mode I've been most frequently referring to here is this second mode, where we go from the wide cells to the rounder, more D-shaped cells. And our modeling that we've done together with Alex Mogilner, together with experimental work, has suggested that really the variation in those shapes is primarily due to the back and forth force balance between actin polymerization pushing on the membrane and membrane tension restraining the actin polymerization. And the short version is cells that have very forceful actin polymerization are able to assume this coherent, wide lamellipodium, and cells that have weaker actin polymerization for whatever reason are the ones that end up in the rounder D shape and also move slower. Now, if that idea is true, then it should be the case that we can take an individual cell and somehow increase or decrease its overall rate of actin polymerization and have that individual cell change across this entire shape spectrum. And so that experiment actually was done by Greg Allen, where the method he chose to change the rate of actin polymerization in the cell was simply to lower and raise the temperature. So here we're going to watch a movie of a cell, and as it moves along, you can see it's fairly slow. It's got this more sort of D-shaped pattern. And Greg is first going to start dropping the temperature. And as the temperature drops, you can see the lamellipodium gets rounder and rounder, and the cell is moving slower and slower. And now at this point, when we get down to just about 7 degrees Celsius, he's now going to start raising the temperature. And as the temperature comes back up, you can see the cell not only goes faster and faster, but it also assumes a wider shape. And so following cells like this quantitatively using a variety of different metrics, what we're able to find is that, in fact, individual cells can also explore this entire range of behavior that we see in the context of cell-to-cell variation. And it's all consistent with the idea that the primary determinant of the shape of the cell, as well as the speed of the cell, is simply how fast the actin is polymerizing. Now, another really fun thing about keratocytes is they have the ability to sense and respond to electric fields. And this is something they actually have in common with many other motile cells. Pretty much any motile cell that you put in a DC electric field will choose either the anode or the cathode and will head in one direction. This was first described for keratocytes by Cooper and Schlewa back in 1986. And Greg was able to replicate this using a setup that he built in our lab to look at motion of individual cells as the electric field was switched from one direction to the other. So in this movie, we see a cell that's moving along in an electric field that is oriented in this direction. You can see the orientation of the electric field and also the magnitude listed over here. And you can see the cell is following that line. Now, when the label turned red, that was when the field was flipped around. And you can see the cell has turned and is now heading back in the other direction. And now, once again, the orientation of the field is flipped. The cell flips back around and now heads back in the direction that it's been told to go. So the cells, obviously, even though I've been emphasizing how good they are at balancing forces across the front of the cell and between the front and the back of the cell, they are able to also initiate imbalances in their forces so that they're able to turn. So Greg Allen looked a little more deeply into the mechanism of how they turn, and he found a couple of interesting things. So for example, if we look at a single turning cell, in this case we're looking at it both in the lab frame of reference, like it looks on the microscope, and then in the cell frame of reference where we've repositioned everything to see things from the cell's point of view, you can see there's a physical asymmetry in a turning cell where the part that's on the inside of the curve, that is the part that's going slower, has this very round shape that we would call decoherent, and that's characteristic of slow motion. And on the outside of the turn, the part that's going faster, has a much more elongated, coherent shape that we associate with fast motion. So this variation that we see, both at the population level and in individual cells as the temperature is raised and lowered, can actually also happen even within the context of an individual cell, where one side can end up being much faster than the other side. Now, there's a number of different things that contribute to this asymmetry, but at this point you won't be surprised that one of the major things that contributes is the left-right distribution of myosin. So I showed you before that the myosin accumulates in these two spots on either side of the cell body. And those two spots aren't always necessarily equal in size. So this is an example of a cell where there's a relatively low amount of myosin in the spot on the left side and a much higher amount of myosin in the spot on the right side. And look in the movie, you can see the consequence of that here with the myosin label. The side that has more myosin is moving faster and is therefore sweeping around the outside part of the turn. Now there's, of course, other elements that also contribute to this turning. There are differences in adhesion, there are differences in traction force, there are differences in rates of actin polymerization, but they all seem connected to one another, and specifically connected to one another through the mechanism of myosin action. So to summarize what we think is going on here, we think as the cell begins to turn, the actin network starts to flow, instead of just flowing straight back to the back of the cell, starts to flow at a slight angle. Because the myosin is carried along on that flowing actin network, the myosin then accumulates at the outside corner of the cell. That myosin is able to contract faster, pull in that side of the back of the wing of the lamellipodium, and help the cell sort of flip around. At the same time, because the myosin is depolymerizing the actin filaments, it's generating a gradient of G-actin such that there's more actin available for polymerization on that same side of the cell where you have more myosin and where you have faster motion. And all of these things we think are able to actually feed back on one another in a positive sense, such that once a cell starts making one of these turns, it actually is able to continue to make that turn in a persistent way, actually for quite a long time, until it's then forced to turn in another direction. So far what I've shown you is that in the context of these very simple steady-state moving cells, myosin in the back of the cell is actually doing a tremendous number of exciting things that help the cell move overall and that help coordinate the front and the back. It's helping to disassemble the network, and it's also specifically contributing to left-right asymmetries that help the cell to turn. Now, the keratocytes are fairly unusual cells. They're unusual in their appearance, they're unusual in the steadiness of their motion. And so it became very natural then for us to ask whether similar mechanisms might be at play in more complicated cells that are doing more complicated tasks. And one of the very interesting cells that's been well studied in the context of motility is the neutrophil, the human neutrophil, white blood cell, whose job is to go after and engulf bacteria that are invading the human body. And you can actually isolate neutrophils from your own blood and watch them crawl around and eat things. It's really very gratifying. But also we have a cell line, a neutrophil-like cell line that is able to behave much like a neutrophil, but that we can also transform and look at protein distributions in moving versions of the cells. So Tony Tsai and the lab decided that it was time to actually try to break out of the keratocyte mold and start looking at motion in more complicated kinds of cells, including neutrophils. And just to show you how dramatic the behavior of these cells is, this is one of these HL60 cells that's been put in a chamber with some Candida albicans, which is a pathogenic yeast. And what you see as the movie loops is the neutrophil starts off over on the right side here and then runs across to this little pile of yeast and is able to actually phagocytose and engulf them. So it really is a very neutrophil behaving tissue culture cell. Now looking at the shapes, they're obviously much more complicated than keratocytes. And putting in labels for the actin, which is shown here in green, the myosin shown in red, and then DAPI to stain the nucleus in blue, what you can see is that the shapes are not only much more variable than keratocytes, but also much, much more dynamic. All of the cytoskeletal elements are drastically rearranging themselves over periods of just a few seconds as the cell is crawling around. So although this makes it a more interesting question, I think, to figure out what is going on in terms of the mechanics and dynamics of this behavior, it's also a much more challenging problem as far as quantitative analysis goes. So Tony so far has been able to work out a number of quantitative techniques to be able to break down this complex motion so we can actually watch changes over time. So for example, he can track the edges of one of these moving neutrophils and then go back and calculate for the cell as it's moving how much of the cell is extending in every time frame, in this case every couple of seconds, how much is retracting, calculate the overall area of the cell as well as the extension of its leading edge and the amount of retraction of its body. At the same time, we can look at labeled proteins inside the cell, and obviously one of the ones we're most interested in looking at is myosin, and look at the overall fluorescence intensity distribution and see how that changes as the cell moves around. And what you'll probably be able to see is that the myosin localization itself is also very dynamic. It's often in the back of the cell, sometimes in these bright spots, but then those bright spots will disassemble, the myosin will become more uniform or will move to a different location within the cell. And tracking all of those things quantitatively over time, what Tony was able to find was that when a cell is speeding up, that the accumulation of the myosin in response to that change in cell behavior happens later, happens about 12 or 15 seconds after the initial movement at the leading edge. So whereas in the keratocytes, it seems like the actin and the myosin were always in perfect balance so that the cells were always gliding forward. In the neutrophils, the story is a little bit different. It seems like the actin is calling the initial shots, and the myosin is then reacting. So as the cell initially extends, it then activates the accumulation of myosin at the rear to pull the back together. So instead of gliding, it's doing more of a sort of inchworm motion. How does this affect cell turning? Well, the same way that Tony was able to come up with quantitative metrics for the localization of myosin, he was also able to come up with quantitative descriptions for the orientation change of the cell and then the left-right asymmetry of the myosin with respect to its immediate orientation. And you can already see the answer actually quite dramatically with this maximal intensity projection, where this is just a low magnification movie of a cell that's undergoing a sinusoidal path. And we're looking now only at the myosin that's accumulating in its rear. And what you can see is that the myosin always accumulates on the outside of the turn. And when it changes its direction, the myosin then changes which side of the cell it's on. So this is actually very reminiscent of the keratocytes, where we saw, again, the myosin on the outside of the turn. Except in the case of neutrophils, again, there's a little bit of a time lag between when the turning initially starts versus when the myosin accumulates on the outside of the turn. So here, too, it looks as if the actin is calling the shots in terms of direction. The flow direction of the actin that's changing then gathers the myosin on the outside of the turn. That then causes disassembly of that cytoskeleton in a way that enables the neutrophil to essentially swing its tail around so that the whole cell is now oriented in the proper direction. So overall, comparing these two stories, if you look at a movie of a keratocyte versus a neutrophil, they seem like they're behaving rather differently. But what we understand when we dissect the mechanics and the dynamics of this behavior is that they're strikingly similar. In particular, I've shown you recent data that myosin accumulates at the cell rear due to this actin network retrograde transport and mediates actin network disassembly in a way that is able to coordinate not only front-rear motion of the cell, but also give you asymmetries that can lead to turning. And both of those things seem to happen in very similar ways in both the keratocytes and the neutrophils. Now, one last little hint I want to leave you with is, as I showed you before with this movie, we can force keratocytes to behave more like neutrophils in terms of changing their shape all the time if we simply change the environment, in particular if we put them on very, very sticky substrates. So you might wonder, can we do the flip side? Can we make a neutrophil behave more like a keratocyte into something that will have a steady state motion where everything's happening at the same rate? Well, it turns out when you take a neutrophil and you confine it to a very narrow channel and then watch it move over time, these guys now are moving at steady state. The speed is absolutely constant. The distribution of myosin is constant, is always found at the rear. And they'll continue to move like this for many tens of minutes without any obvious changes in terms of their overall shape or overall behavior. And taking the same movie and now making a chimograph of it where the time is moving from top to bottom and each one of these slices is an individual frame of this movie, you can see really how constant the speed is over time. So not only can we force keratocytes to become more crazy and change their direction like neutrophils, we can also force neutrophils to behave more in a steady state like keratocytes. And moving forward, I think the combination of our ability to both measure and manipulate these different kinds of motile cells will help us to understand the general principles that govern motility for all animal cells that use actin polymerization to drive their movement. So as an overall summary, I think the main point here is that actin and myosin have to cooperate in order to make cells move, not just in order to generate force, but also in order to do things like steer them and determine their shape. And we found that myosin plays actually several very unexpected roles at the rear of cells, not just contributing to contraction as we might have expected, but also contributing specifically to actin network turnover and to asymmetries that lead to cell turning. And overall, we've been very surprised by how similar the mechanics are between fish skin keratocytes and human neutrophils. So obviously, there have been a lot of very talented people who contributed to the work that I've just described. And I've listed here the many members of my group who have contributed to different aspects of cell motility projects over the last 15 years or so, and also our wonderful collaborators. And I'd particularly like to mention in this context our very productive long-term collaboration with Alex Mogilner, who has done a lot of the quantitative physical modeling that has driven the thought processes behind our experiments. Thank you.